Good morning. I will be presenting about the gut microbiota of the honeybees, and my mentor is the very cool and awesome Magdalena Warren. So bees are a really big part of our agriculture. They pollinate about 1/3 of the food that we eat, but they are in great decline. About more than 50% of North America's native bee species are in decline, with 1/4 at the risk of extinction. Um, And by understanding the interactions between their gut microbe and the bee, we could find better ways to protect them. So I was involved in two projects of my mentors. One of them is the strawberry project, which relates to pesticides. And the other one is the apricot project, which has implications for in protecting the bees against pathogens. Due to time limit, I will only be talking about strawberry projects, but if anyone has questions, please ask. For the strawberry projects, we are trying to find how agricultural intensity affects the bee microbiota, and my specific focus is to prepare the fungal library for sequencing. So before we hop right in, I want to talk about the anatomy of the bee guts. So the bee gut or the stomach is made of the crop and the intestine. You can see the crop kind of looks like a bubble. It is where the bee stores their nectar. And to the right, you can see a diagram by Moran in her 2016 paper where she suggests that the microbes in the crop are mostly environmental, while the microbes in the intestines are core microbes, meaning these microbes are present in the gut of all bees. And the strawberry projects, we specifically look at how agricultural intensity affects these gut microbes. And to do that, we take the microbes from the intestine/crop and we perform DNA extraction on it to get all the DNA. And then we perform PCR to amplify the target gene. In my case, I amplified the ITS gene so I can find the identity of the fungi. And then I did gel electrophoresis just to make sure that the PCR works. And if the gel comes out good, I would continue on with DNA purification to get rid of all the extra stuff we don't need and just have just to have the DNA, the cleaned up DNA by itself. And then next we would submit that to get it sequenced. And once we have the sequences of the DNA, we can find what types of microbes, what types of fungi, or what types of bacteria are in the crop/intestine. And from that, we can do a community analysis. These are some of my gel electrophoresis results. So the gel electrophoresis, it makes sure that there's no contamination and makes sure that the PCR works well. And if I may point out, the negative control, it's clear. This white spot here is the primer, which is normal, which is the clear negative control is what we want because it shows that it means that there's no contamination that happens while we were pipetting the DNA. And that's good. So I did not finish the fungal community analysis, but we do have preliminary results that we did on the bacteria. So this is the bacterial community analysis. And here in blue is the bacterial community in the intestines for the Monterey farm. It is the most agriculturally intense farm out of the 5 farms that we have. And if we go from the most agriculturally intense to the least agriculturally intense, we would have the True to Earth farm in purple. And then next we would have the Berrylicious family farm in orange, Crystal Bay farm, and finally the Life Earth farm, which is the least agriculturally intense. And When we go from most to least agriculturally intense, you can see that there's a slight shift to the right, which is not what we would expect, because we would expect all the data to be on top of each other. We would expect the data to be the same because the microbes in the intestines are core microbes. So all the bees have them. So we would expect them to be the same, but they're not. And if we organize the data in terms of conventional farm versus organic, conventional being that the farm uses pesticides slash herbicides, and organic being that they don't, we can see that there's also a significant difference, which again, we would not expect. We would expect it to be the same. And here it's basically the same, but it's looking at the bacteria in the crop. Although that it is the data is different and it corroborates our previous data, we should keep in mind that the microbes in the crop are not as stable as the ones in the intestines. So our next step would be to finish the fungal analysis. And once we have that done, we would compare that with the bacterial analysis. And that is to see the patterns of interaction between bacteria and fungi in the honeybee guts. For example, an increase of one species of bacteria could correspond with a decrease in another species of fungi.
You have about 30 seconds, so if you could wrap it up, please. Thank you.
And next, we would perform metagenomics, which is to analyze the function of the genes. So some key skills I gained was working with DNA, and one of the best aspects of my internship was meeting the people of the Fukumi lab and my mentor. And I like to say thank you to the funders for funding the project, and thank you to SLI and my mentor Maggie for making my summer one of the best summer I've had. Thank you.